Good morning and welcome to the Skye Bioscience expert panel. At this time, all attendees are in a listen-only mode. A question and answer session will follow the formal presentations and fireside chat. If you'd like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player. As a reminder, this call is being recorded and a replay will be made available on the Skye Bioscience website following the conclusion of the event. I'd now like to turn the call over to Puneet Dhillon, Chairman and CEO of Skye Bioscience. Please go ahead, Puneet.
Hey, good morning. Thank you, Tara. President and CEO, Paul Grayson's the chairman. But good morning, everyone, and welcome to today's call. Today's about framing how to interpret the CB1 phase 2A top line, and what we're going to be talking today about what is meaningful, how we're translating the clinical readout into the next steps. But before we begin, I want to remind everyone that we're not previewing any clinical results today, but still we're going to be referencing some new preclinical data that we just announced today. And we're going to remind everyone of the cautionary note regarding forward-looking statements. Regarding the agenda, I'll start off with an introduction. I'll turn it over to Dr. Puneet Arora, our Chief Medical Officer, to cover the brief science and clinical update. And then I'll come back and talk about what success looks like at 26 weeks. And then we're going to follow it with a KOL discussion with our expert panel, and then followed by audience Q&A. So we're very thankful for the expert panelists today. Today we have Dr. Sean Wharton, who brings deep clinical experience across anti-obesity medications in the real world and clinical perspectives. Dr. Kevin Cannon is also an endocrinologist and expert in obesity, as well as a CB1 PI, and can speak to the trial conduct and interpretation as well. And then Dr. Marcus Gonsalves is a physician endocrinologist and biomedical researcher at NYU Langone, and he bridges the intricate biology of the endocrine system as well as human metabolism. And I'm going to be moderating. Dr. Puneet Arora, our Chief Medical Officer, will cover the science and study design. So over the past year, we've been on a warpath to prosecute a validated CB1 pathway with a novel peripherally acting allosteric antibody, which is Nimacimab. And on the left of this screen is the evidence we've built, and on the right of the screen is the new data that we're sharing today. Just to orient the listeners here, on the left side is really about the foundation. So if you think about this across multiple species models, especially the DIO models, Nimacimab reduces fat metabolism while preserving lean mass. We're showing data now that modulates key hormones like GLP-1, leptin, resistin, and we've also shown weight loss durability after dosing stops, as well as dose-dependent weight loss. So this is consistent with a mechanism that really resets the metabolic homeostasis. We also have shown data that highlights improved glycemic control, lowering fasting insulin, glucose, overall better tolerance, plus liver and lipid benefits with reduced steatosis and serum cholesterol. Importantly, the other data that has really been supportive of a unique mechanism here is the inflammation and fibrosis signals moving in the right direction. And then when we add Nimacimab to the incretins, we see a greater additive weight loss preclinically. So mechanistically, it's really this distinguished molecule being a negative allosteric modulator of CB1, and it's designed for this peripheral action. And that's also been helpful from a safety perspective, looking at multiple NHP biodistribution studies that highlight that it's unique in terms of being able to remain in the periphery. Today, we're adding two fresh new data sets. One is dose titration versus monlunabant, and there the data is supporting Nimacimab compares favorably on treatment, and again shows limited post-treatment rebound across doses. And for those that want to really dive into the details on the new data, we've posted 4 new videos today on the Spotlight page with slides and a voiceover by Dr. Chris Twitty, our Chief Scientific Officer. So I'm not going to get into all of the key takeaways in my intro, But the additional data point, so there's the monlunabant data point, and then there's a repeat DIO study where we're looking at nimacimab alongside of tirzepatide, evaluating a combination with suboptimal and optimal doses of tirzepatide. And the headline there is that we've shown we're driving up to 47% vehicle-adjusted weight loss with minimal rebound profile, and that again supports this maintenance potential. On the just looking ahead side, on the clinical front, the phase 2 proof of concept readout is expected late Q3, early Q4. And in parallel, what we've been working hard on is preparing for the phase 2b, which we're going to refer to as CB1 2. And that's to evaluate multi-dose and dose frequency to ultimately set the dose or set the stage for the phase 3 dose. We've also, as you know, we've highlighted this year, we've established a very robust R&D a preclinical program with this human CB1 knock-in model, and we have multiple data points coming out of that effort on a steady basis. So we're going to continue to have additional preclinical readouts into 2026. And lastly, I'll just mention that we've just recently completed the last patient visit in the 26-week treatment phase. So we're fully enrolled in the 26-week treatment phase, and that's done. And we also fully enrolled in the 26-week extension. So now that'll enable us to get to the 52-week data point for those patients that are participating in the extension study. And Dr. Aurora will cover that in more detail. But in terms of data points, we're guiding for the extension readout in Q1 of 2026. So just to quickly orient the listeners on the molecule, nimacimab is engineered for peripheral restriction. It's significantly less in terms of brain penetration when we compare against small molecules, which really speaks directly to the safety concerns associated with the historic CB1 approaches. Mechanistically, it's a negative allosteric modulator, so it's retaining the potency even when you have endogenous ligand competition like 2AG and AEA. And this combination of having a peripheral CB1 engagement with really this allosteric control, I would say again underpins our safety and tolerability first TPP or our target product profile, and we're really able to then preserve efficacy with allowing for potentially an adequate therapeutic window, meaning that we have a very favorable PK/PD profile for dosing because of this unique difference from small molecules. Again, we've covered this in corporate updates, but just to orient the listener here, there's really four mechanistic pillars that we've been highlighting. Peripheral CB1 modulation affects appetite hormones, so leptin and GLP-1. It improves glycemic control, so fasting insulin and glucose, enhances lipid metabolism, things like steatosis and cholesterol, and it reduces inflammation and fibrosis. And at the top line readout, we are looking beyond weight loss. So many of these biomarkers will be evaluated, and this would provide really strong mechanistic support for the antibody. Okay, so we've recently created this slide, and really the purpose of this slide is to just highlight that there's plenty of white space if we focus on the issue of tolerability. A few percentage points in terms of improving GI, and this slide is kind of highlighting one GI tolerability data point that's consistent across incretins, looking at nausea. And if we are able to have any improvement there, you can see that it really puts nimacimab in the spotlight in terms of being able to show a differentiated mechanism that can improve improve the GI tolerability concern. So for us, there's kind of three ways of really looking at this. There's a value here from a monotherapy value perspective, looking at tolerability as an improvement, where we can aim for semaglutide-like efficacy with a much better day-to-day tolerability. There's also potential here for combination, and we've covered this preclinically, but we're looking at this carefully in the clinical setting as well. Because CB1 is a differentiated mechanism to the incretins, we are targeting for a cleaner tolerability profile, and combination really becomes feasible. And that's something that we're going to be watching for in the clinical readout. And then the other side of the market is this maintenance side. And what you're going to hear today is better tolerability is what keeps people on therapy long term. And that's really the foundation for long-term use and maintenance of maintenance of weight loss after patients come on incretins or any other GLP-1 initiation or discontinuation. So it's really the biggest the biggest barrier, we think, is the drop-off rate because of GI burden, and a cleaner profile really highlights where nimacimab can really capture a big segment of the market. And that's just a setup here in terms of highlighting the three kind of practical use cases that are emerging. This year, we spent a lot of time speaking to clinicians. We've done a survey of approximately 60 different endocrinologists and PCPs, and it's evident that there's a really distinguished market here for monotherapy, combination, and maintenance, where physicians have highlighted an unmet need around tolerability and chronic use. And what we're highlighting here is that there's an opportunity for a more tolerable regimen that supports the chronic therapy. And this includes the GLP-1 intolerant, or patients that have limited response, or those that are even looking for needing additional weight loss or maintenance after they initiate GLP-1. So our TPP is really built to address those realities, and we feel that our molecule does an excellent job on that. Now, I just wanted to kind of look at some of this overarching DIO evidence that's been built, and this is another visual here that really is emphasizing three important points. One is that when we look at DIO studies, they really inform the translation to human data directionally. So when you look at across obesity mechanisms in any rank order, in terms of different mechanisms, or looking at combination in DIO, they've tended to translate directionally to human data. Number two is that CB1 is a very clearly validated biology. If you look at across multiple different CB1 mechanisms, including now with nimacimab, this peripheral antibody, all the different data speaks to robust DIO weight loss and an overall favorable metabolic signals. It's really supporting the mechanistic validity of the class. And the third point is specifically on nimacimab, where we've been able to show a consistent and very versatile molecule in repeat studies, and showed now dose-dependent monoefficacy, combination additive weight loss, as well as the maintenance utility. And that's exactly how our TPP is designed, right? So we're looking at monotherapy, combination, and ultimately maintenance. And when we look at this, from a high visual point of different data points, it really speaks to the robustness of what's been happening in the preclinical side. So today we're obviously not going to go through every single thing, but I've highlighted a few areas that we're covering. And the 2 points that we made in the press release today is favorable comparison to monlunabant. And then we also highlighted a combination with tirzepatide, evaluating rebound and maintenance after withdrawal of drug. And on that note, I'm going to turn it over to Dr. Arora now to walk us through some of those highlights.
Thank you, Puneet. Now, you've all heard from Puneet the important potential role an antibody to CB1, such as nimacimab, can play in the treatment of people with obesity and metabolic disorders. And this is true as a monotherapy, it's true as a combination, and equally important, it's true in maintenance of weight loss after initial weight loss has taken place, now that we know that there are effective agents out there that can cause the weight loss. I'm going to take you through some really exciting data that underlines the strong science and the biological rationale for the potential of nimacimab. And we're going to look at these models that we've created. These are DIO mice. They're humanized mice in that there's a human CB1 receptor that's been knocked into these, so we can use the antibody nimacimab itself. And in this first set of experiments that you're seeing in front of you now, we're looking at monotherapy. So we have a vehicle and then three different doses of nimacimab. So we're going to see a dose response for monotherapy with nimacimab. And equally important, we've added into this monlunabant. Now, you're all familiar with monlunabant. It's a small molecule CB1 antagonist being developed elsewhere. And it has clinical data. So it's part of this new generation of CB1 antagonists. These mice were all treated for 24 days, which in mouse terms is a pretty significant period to be able to see the effects of these agents in terms of weight loss, and then followed up for another 24 days to make a total of 48 days. And in this second phase, there was no intervention. So we can see what happens when treatment is ended. So these panels all show you absolute weight on the left side, and then vehicle or placebo adjusted weight loss on the right side. So I'm going to focus on the right side, and you can see the flat line for the control. And then you can see a dose response in the three NEMA doses, nimacimab 75, 240, and 750, with a very significant weight loss all the way up to 24.2% in the 24 days with the highest dose of nimacimab. In the green, you can also see monlunabant, and you can see that monlunabant, compared to nimacimab, tends to flatten out in terms of its response relatively early, and the final response of 13.9% was just above what we saw with the two initial doses of nimacimab. So we saw some very favorable data in terms of total weight loss in 24 days with the nimacimab doses as compared to what we are seeing here with monlunabant. Now let's look further and understand what happened when these cohorts of mice were withdrawn from therapy. So we can see up there in the black line the control mice, and you can see that they end up gaining some weight back again. There was some initial weight loss in these mice. Monlunabant, we had already seen, had some flattening out, and then once the drug is withdrawn, there's a relatively quick uptake in terms of weight regain. Now, it's interesting, we've learned this from patients over the years, that weight regain after you've lost weight, and this is especially true when we put people on diets or suppress what they eat, ends up at a point not where you necessarily began your weight loss journey, but where you would have been if you had never undertaken that journey. And this becomes important as we go on, and I'll point it out again. But here you can see that monlunabant is back to approximately where the control is. On the other hand, with all three nimacimab doses, you see that nimacimab tends to have a more prolonged mitigation of this weight regain and a relatively flat period where there is no weight regain initially till about day 33 or day 36, depending on the dose, and then some weight gain after that, as you see with any weight loss, to the extent of about 4%. So it appeared here that nimacimab drives a more durable weight loss and a relatively minimal rebound as compared to monlunabant treatment in this model, which bodes very well for maintenance of weight loss after treatment. So let's look further, and another experiment that is designed similarly in two phases, and this time we're using a vehicle and the middle dose of nimacimab that we saw, but we're comparing it to cohorts where 2 different doses of tirzepatide are now being administered. So there is a low dose and there is a maximal dose of tirzepatide. And then, really exciting, we have a combination dose where we're giving the lower dose of tirzepatide in combination with nimacimab. After the 24 days of treatment, the nimacimab will be withdrawn, but the lower dose of tirzepatide will actually have a comparison between some mice that will be withdrawn from treatment and others that will then have maintenance therapy with nimacimab. Similarly, the high dose of tirzepatide, these mice will also move to nimacimab treatment to see how we can maintain the weight loss. And the same will be true of the combination. So let's take a look at what we found here. So once again here, the black line on top represents the adjusted control. And you can see below that, the nimacimab, which looks similar to what we had seen before, about a 21% weight loss over 24 days. And then the 2 doses of tirzepatide, as you would expect, they have about a 30% and a 39% weight loss at the end of the 24 days. But the really exciting thing here is that when we combine the lower dose of tirzepatide with nimacimab, then at the end of 24 days, that's where we see the maximal weight loss, more than what we are seeing with the maximal dose of tirzepatide, up to about 44%. And it's always equally interesting to me what the trajectory of the weight loss is. And the suggestion here is that this weight loss isn't done, especially in the combination treatment. And in this arm, it appears that if we continue to treat, there may be more weight loss yet. So going forward and looking at what happened after that, the control arm, as you would expect, just continues to gain weight just in general through the study. With tirzepatide, we're looking at the lower dose of tirzepatide, and we're looking at the mice that had withdrawal of treatment. We can see a very rapid weight gain. And as I had mentioned to you before, the weight tends to go back to where you would have been if you had never taken any of this treatment before. But in comparison to that, the nimacimab rebound looks very different. For one thing, up to about we had seen this previously, up to about day 36, the weight tends to remain flat, and the trajectory of the rebound is slower after that as well. And we'll go on to see more with that. But let's look at what happened to the other cohorts here. So we now can see what happens when the low dose of tirzepatide, instead of being withdrawn, was continued with nimacimab instead as a maintenance dose. And you can see here a significant mitigation in the weight regain that took place. The same is true in the circles with the red outline for the high dose of tirzepatide, followed by nimacimab. And finally, right at the bottom, you can see, and we've already seen that the combination does really well in terms of maximal weight loss, that if you follow that up with nimacimab instead of withdrawing treatment, there is a significant mitigation of that weight rebound, and weight can be maintained. You can see the flattening coming out of that with nimacimab. So switching to nimacimab treatment does limit rebound and nimacimab here shows significant potential as a maintenance therapy. So finally, this is a more complex experiment. We're adding more arms. You can see that we're now dividing up the high dose again into maintenance with either vehicle or with nimacimab. But what I'm going to focus right now with you today is on this particular arm in the light blue color, which is listed second, where we have a pair-fed set of mice to nimacimab. And what we're trying to show here, or we're trying to answer the question of what the role of suppression of appetite with nimacimab is in terms of this weight loss. And this question comes up all the time because we talk about a peripheral action, but the mice who take nimacimab, based on this peripheral action, still have a decrease in their food intake. And the question is all the weight loss that you see with nimacimab only because of this decrease in food intake? And we can answer that by feeding these pair-fed mice in the same way that the nimacimab-treated mice eat. So let's take a look here at what happened, and you can see pair-fed mice above and nimacimab right below that in the lighter blue and the darker blue. And you can see that when you pair-feed mice with nimacimab, initially the trajectory seems to be similar, but very quickly with pair-fed mice, it flattens out, and there is even some regain of weight back again, whereas in the nimacimab cohort, weight gain continues all through the time that we see treatment here for 24 days. So it's clear that nimacimab enhances weight loss all along here, not just because of the decrease in appetite and the decrease in intake is seen even with these pair-fed mice, but because of other mechanisms as well. And this is what happens when we discontinue. The pair-fed mice just continue along and end up right where the control arm is, whereas at day 35, when the nimacimab treated mice are seen, there is a flat trajectory, and in fact, we're not even seeing the beginning of weight regain here. So this highlights that nimacimab-driven weight loss is not a result of caloric deficit alone. So having gone through some of that really exciting new DIO data with you, I want to talk for a couple of minutes about what we're doing right now. You're all aware that we are in the midst of the phase 2A CB1 trial in patients with overweight and obesity. This trial has a 26-week treatment period, and all patients have now completed the 26 weeks of treatment, and we're looking forward relatively soon to having top-line data from this trial. Just to revise the design of this trial with you, we have 4 arms in this trial. The randomization is 2:2:1:1, which means that the major part of this trial is nimacimab versus placebo. And the second smaller part of the trial is where all patients get Wegovy, and we're looking at a combination of Wegovy with nimacimab versus Wegovy with placebo. So we will also have data from this study on what happens when you combine treatments with Wegovy, in this case, which is semaglutide, with nimacimab. All of these cohorts are being treated for 26 weeks in the study, and as per the original design of the study, they would then get a 13-week follow-up from the last dose of nimacimab to week 39. Now, we have now introduced another arm in this study, which is an extension to the study to get further data out of this. Now, we did include the extension to the study in a somewhat belatedly, and by the time the study was set up and all the sites were activated for the extension, we had about 57 patients out of the original study that were eligible to go into the extension. And we have now completed enrollment for this extension, and 43 out of the 57 patients have enrolled, 19 in the combination arm and 24 in the monotherapy arms. In order to maximize the number of patients who would be eligible, we allowed patients who were in the combination arm to roll over to the extension up to 4 weeks after the last dose of their treatment, and they would then get 26 weeks of treatment further. Since they're already on an active drug, we can keep the combination patients on blinded treatment. So these patients will continue either on semaglutide plus placebo or semaglutide plus nimacimab. And that's the data we'll be able to present to you 26 weeks from now, approximately sometime next year. The monotherapy arm, on the other hand, has a placebo arm, as you all know. So we're going to give all the monotherapy patients that go into the extension study open label nimacimab to give everyone the opportunity to get active drug. And in the same way as the combination arm, they will get an additional 26 weeks of treatment. So we will have up to a 52-week data readout over here. Now, the monotherapy patients who started later in this study were allowed to roll over into the extension if they were still enrolled in the study and had completed 26 weeks of treatment, which means they could be as long as 13 weeks outside of their treatment and still be eligible for extension. Okay, I'm going to hand it back to Puneet to talk about what we should expect from this phase 2A CB1 trial.
Thank you, Dr. Arora. Okay, so let's shift to this top line expectation side. So key point here is that the success band that we're looking for is approximately 5 to 8% placebo-adjusted weight loss at 26 weeks. We've also indicated that if we're looking at 8% or placebo adjusted, that's the upside scenario in terms of representing really the high end of the proof of concept study outcome. So the things we're watching for here is slope. Everything ultimately ties back to the TPP in terms of the efficacy signal that we're looking at. So one of the key things that is an important consideration is the GI tolerability side as well. We want to obviously highlight a very durable weight loss with a slope that's continuing to come down. We're going to evaluate. It sets us up well for the 52-week view, and then ensuring that we continue to see a really strong safety and tolerability side. When we look at combination, the expectation here is to have an additive weight loss to GLP-1. GLP-1s have shown about 9% to upwards to 11% at approximately the same time point. So we want to see anything additive on that. But importantly, again, a path to better tolerability. As we've highlighted before, closely watching nausea, vomiting, diarrhea, or discontinuations related to any of those issues. So ultimately, having better adherence and better combination usability with better or a more favorable tolerability profile in combination is a huge win for nimacimab. So the idea here is to support a path to double-digit weight loss. This is unprecedented territory, so we are going to be the first readout for showing in human CB1 and GLP-1 data. So we don't have a true comparator, but the idea is to have additive weight loss with a better tolerability profile. The next kind of thing is obviously the issue of GI tolerability. We're looking at that closely in mono and combo. As I said, looking at all three common tolerability concerns. And the expectation there is a lower GI burden at the end of the day with our mechanism. We have really good evidence of this from phase 1 and the phase 1b. So we want to be able to replicate that in a larger and longer trial. The idea, is not only to have a best in class, but potentially a best in field tolerability profile. Next, I'll just move to safety. So at the 26-week time point, clearly that the biggest thing that is being watched in this mechanism is neuropsychiatric concerns. We have highlighted time and again our unique peripheral restriction and the fact that we don't see any of the same magnitude of brain exposure as other historical compounds in this class. So the gate for us in this expectation setting is really no neuropsychiatric concerns. We think that that's the cleanest and most best way to have adoption of this particular mechanism. And this is why we bet on this from day one, is because nimacimab had a very unique profile based on clinical data. So clean safety is really an important gate for us going into phase 2b for the antibody. And then finally, we are looking at body composition and biomarkers. So we'll look forward to highlighting that. The body composition trend that we're expecting is more of a preferential fat loss. And on the metabolic side, we're looking at a range of different metabolic biomarkers that really support the peripheral CB1 biology. I'm going to just focus in on tolerability. So there's really kind of three important strategic looks here, or things that we think are important for unlocking kind of that view on tolerability. One is that we're looking to see a real strong monotherapy differentiation here with semaglutide-like profile in efficacy with materially better tolerability, and that makes it a really compelling monotherapy. So we don't need to beat GLP-1 magnitude to win commercially. We just need to be able to really uh, be able to show that differentiation where you can rescue those patients and keep them on drug longer to have better sustainable weight loss. Number two is the combination potential, and that really makes for a more tolerable GLP-1 combination. And we feel with that, based on this preclinical evidence, that really can target higher weight loss and potentially better body composition without compounding any of the GI burden. So this is something that we think that the prior combinations with non-incretins really have struggled to achieve. And this is a differentiation in terms of the nimacimab mechanism being able to really have this broader metabolic reset. And third is the persistence and maintenance component. We believe that better tolerability at the end of the day is really the underpinning for maintenance role, and that's why that's one of our top line priorities. It's looking at safety, including neuropsychiatric concerns, the GI tolerability, and these other things that we've been talking about, including the trajectory and the combination side. So that's essentially kind of the three important points we want to make in terms of a case for why we expect to have better tolerability and why that matters. Obviously, that's something that we're seeing how that translates directly into the top line readout priorities on the right side of the screen in terms of safety, watching for GI, looking at some of the mechanistic quality side, and then, as I mentioned before, the efficacy signal and the trajectory. With that, we'll open it up now for a discussion with our panelists. So I'm going to first ask kind of the burning question to Doctors Cannon and Warden. So the first question we wanted to ask was for a first-in-class peripheral CB1 monoclonal antibody like nimacimab, what's clinically meaningful weight loss range at 26 weeks, and how much does that slope matter?
Great. So who wants to take that first? Kevin, did you want to take it, or did you?
Start, and then you can. Okay, yeah, go ahead with it. Thanks, Sean.
Yeah. So, so I think if we're looking at the 26-week mark, it's a little challenging to say exactly how whether the amount of weight loss at that point, or even the slope, is indicative of what's going to happen at the 52-week mark. What we've been seeing recently is that that doesn't exactly tell us what happens at the end of the year. So it's been a little bit disappointing that we weren't always clear that getting to a 8% or 10% or even further was going to dictate a greater amount of weight change at the actual 52 mark range. So I would say that you've got to be in the ballpark. Got to be there. You've got to be in the ballpark between that placebo-adjusted 5 to 8%, or just overall weight change about 8 to 10 percent range, with a trajectory that the slope looks like it's continuing to come down. Whether that flattens out after those 26 weeks and starts to flatten out at 46 weeks, 48 weeks, or keeps on going, is to be seen at the 52-week mark. But so I don't think that you can count your chickens exactly at that stage, 26-week. But you got to be a player. You got to be within that ballpark, and it looks like nimacimab will be in that ballpark.
I agree 100% as far as being in the ballpark and the trajectory. The one thing that I'm very much focused on, I think kind of more the population is really starting to take into focus, is that lean muscle mass preservation. And mechanistically, this is exciting that nimacimab is going to help preserve this lean muscle mass, because the quality of we're going to start looking more and more about the quality of weight loss and not just the numbers. And so again, the nimacimab with the mechanism of action and the preclinical data leads me to be pretty excited as far as what we're going to see with those numbers.
Great. If we kind of drill into that further, so if we're starting to see in the higher band of those ranges that Dr. Warden pointed out, do you think that that's competitive to semaglutide? Like, what would really make it better in your opinion, if you're comparing it against current standard of care?
From a lean muscle mass preservation?
Yeah, like lean muscle mass. I think you highlighted. Is there any other things that you're kind of looking at? I've mentioned kind of tolerability ad nauseum today in the opening remarks, but just wanted to get your perspective.
I mean, honestly, yeah, you've highlighted all of the highlights. The butterfly effect from GI tolerability. It's not just maintaining on medicine, but as well as far as when you have that GI tolerability, you're able to allow the patients to really focus on the quality of calories intake as opposed to just the quantity. When you're not having good GI tolerability, they're going to kind of reach to anything that's going to get some calorie intake. And a lot of time that what they're reaching for is not the quality that you're wanting them to focus on. And so again, GI tolerability is going to allow you to really allow them to focus on protein intake and really the quality that you want. And so again, the butterfly effect from, just simply the statement of GI tolerability, that has a lot of positive butterfly effect that goes with that in a structured weight loss program.
Yeah, and I think I would echo the fact that the competitive aspect, yes, is there a competitive aspect to semaglutide right now? And I would say that maybe there's not necessarily need to be a competitive aspect against sema. The thing that we're really seeing is that even with medications that are having higher weight loss than semaglutide in that step 1 trial, it was a big deal trial getting to 15%, but there's some medications that are going to be at 20%, but they're not as good a molecule. If you're having greater side effect profile, if you're seeing a bunch of lean mass loss, if you're seeing a inability to tolerate it with the right type of dosing, the right way, you have to take it with certain you can't take it with certain medications. There's a whole bunch of other things that sema seem to lock on to have an okay tolerability, a pretty good weight change, and that is landing it in a decent spot. So competition against a molecule like that has to have all of those things. Has to have the tolerability profile, has to have the weight change profile, and you may not just be competing against the weight change. You have to compete against just about everything to try to have that competition. But I don't even know if it'll be sema by the time this is out, whether you'll be competing against. It'll be amylin, it'll be other molecules.
Right. Okay, so let's talk about the other burning question, and this is maybe directed towards you, Dr. Wharton, or anyone on the panel. But I'm just saying from your experience on the small molecule side that you might have a unique lens on this. Is there any amount of neuropsychiatric side effects that would be acceptable from the medical community for the CB1 class of drugs?
Right, and the answer to that is probably no. So going back in history to try to understand where all this came from. So as a cannabinoid antagonist, you're antagonizing the endocannabinoid system. The endocannabinoid system is there in our body to help us to get hungry. We get the munchies whenever you smoke up. That's cannabinoids, right? And we have our own cannabinoid system that tells us it's time to actually eat. So you block it, and then you start to block hunger. That's the way this worked. But also at the same time, we saw a lot of great peripheral things happening, not just blocking the hunger, but we saw that a lot of fat loss was happening, a bunch of other things. But the older molecules got into the brain, blocked the cannabinoid system, and that seemingly should be okay, or maybe it's not okay. We just didn't know what was going to happen. And what happened was not good. Right, there was a lot of psychiatric components to that when it got into the brain. So the idea here is, can you have a molecule that doesn't get into the brain, which is what the monlunabant is trying to do, not have that component of getting into the brain, but still maybe does, and maybe there will be some side effects because it's the same type of drug. It's called a band, just like the Ramona bands were. So they're the same category. Whereas here with nimacimab, we have an antibody that's big and doesn't get past the blood-brain barrier, stays in the peripheral system, activates leptin, GLP-1, and all the other things that do get into the brain. So leptin goes into the brain, the GLP-1 goes into the brain, all those things get into the brain to decrease the actual hunger, but the antibody doesn't go into the brain to cause those endocannabinoid problems. So I think this is a unique molecule from that standpoint. And the hope is that because it doesn't penetrate, it doesn't cause the cannabinoid problem at that receptor. So you really have to go back to the big ideas of why this is here, why we're talking about this. Otherwise, we don't have much to talk about. You really got to go back to the history. And the history is that no psychiatric side effects will be really tolerated.
I agree
Okay.
100%.
Yeah, thank you for that. That's a really good explanation. Just to switch into the combination view, so we made this remark in terms of expectation setting around additive weight loss is what we're expecting in the combo. What would you say from a clinical perspective qualifies as meaningful additive weight loss in the GLP-1 combo at top line at the 26-week view?
I mean, pretty much exactly what we said previously. As long as you're in the ballpark and that trajectory, as long as it's an increase and that trajectory is continuing on that downward slope at 26 weeks, I think we can all be very encouraged.
Yeah, so, and we did a study looking at the addition of naltrexone bupropion to the GLP-1s, and this was in people with type 2 diabetes, and we got an extra 4% weight change. Now, people do add this as a combination medication to the GLP-1. So the big deal about the naltrexone bupropion is it's not a GLP-1. The big deal about CB1 antagonists is they're not GLP-1s. That's a very important line because it means you can combine them with the GLP-1s. So it's not just about combining it to the highest dose of a GLP-1 and trying to push the weight down further, like 4 or 5%. But can you combine it with a low dose and get into those teens? Can you get into 17%, 18% if you're combining it with a low dose of the GLP-1 that would normally get you maybe about 8%. So you're seeing that you're adding another doubling of it. So we don't just want an extra 4 or 5 of the top dose. We want almost a doubling of a low dose, getting to the ranges where we're at that 17 to 20%. 20% is where we would like to see these newer medications land. So we've got an actual goal here.
Right. Okay, this is a lot. So, okay, there's one another kind of important. If we dive a little bit deeper into the mechanism difference, but before we bring that back to you guys, actually I'll let you guys stew on this maybe, and then I want to just switch to Dr. Gonsalves, and then we can come back to the clinical side. For Doctors Cannon and Warden, I think one of the things that we were curious about is how CB1 and GLP-1 can really be positioned to work where other non-incretin add-ons, have maybe not been so successful. Like tolerability has been one area, but more, talking about from the differentiation as the orthogonal mechanism. So that's something that we want to come back to you, but maybe to set up the clinical discussion, we can talk to Dr. Gonsalves for a moment here on how investors might kind of translate all of this data that we're sharing today and have shared this year regarding the mono combo and maintenance signals we're starting to see observed in the DIO models and how that's kind of translatable. And I think the second part of that question, Dr. Gonsalves, is really maybe you can explain kind of the difference in the rebound curves between nimacimab, tirzepatide, and monlunabant, and we can flash up any slides you want.
Yeah, thanks so much. Yeah, the data is very exciting, looks really promising. lot of people usually have concerns about using the mouse models to predict or mimic human physiology, but in this setting, it's been very valuable over the years to use the DIO model to mimic what we eventually will find in human subjects. It's not perfect, but it definitely can give us a flavor and a taste for what we will expect, and that has certainly been the case for the incretins as a field. Many of the things we are seeing in human patients were first observed in the DIO models. The major clinical problems I see today in this space is really outside of access, is that the majority of people, 2/3 or greater, are off incretins at 1 year, and that about 2/3 of the weight come back once you're off the incretins. So those two specific issues are completely addressed by the data you presented today. It appears that the addition of this combination, is not predicted to exacerbate any AEs given the mechanism of action, which is important. And we also need effective drugs for that rebound state. And as you can see from your data, the combination does look like it's giving you increased efficacy in terms of the weight loss. And the rebound data is quite impressive. You can go back to those curves where you see just that rapid orange line from tirzepatide go all the way back up even above the starting weight, which is really dramatic. And that a degree of rebound is cut pretty drastically, 50% or more by switching to Nimacimab, which is predicted to not have the GI toxicity. The other point about these rebound, so why is the rebound being mitigated? I see two major themes here. One, I guess the more obvious one is maybe the antibody is just still bound there. It's cleared from the circulation, but it's still bound in the tissues and acting. That's one possibility. And the second, I think, more intriguing possibility is that there's been some sort of metabolic rewiring that's occurred. And some options are maybe there's improved leptin sensitivity, which has also been seen in this space in prior studies. The CB1 receptor could be modulated itself with persistent negative signaling. Maybe it's just downregulated, and all these things lead to this improved state where, the drug is no longer needed to see this beneficial effect.
Yeah, thanks. That's really helpful. Sorry, we had a bit of technical side on the slide. So you were saying on the tirzepatide combo, the curves of the rebound were kind of important to highlight. So looking at this,
yes,
what you're referring to here. Okay.
Yes, exactly. Look at that. I mean,
yeah,
30% comes back over and above the 0 point. It's like they somehow remember what the placebo group is doing and then go all the way back up, which is, a bit odd to me, but that has been shown previously. It's not unique to this study. And then the Nimacimab, you see, if anything, only 25 days later do they come back to their initial baseline, but there is none of this over and above the weight regain that you see with the tirzepatide. I also just wanted to highlight too, which I forgot to mention, is the pair-fed feeding data. It does highlight that there is something new happening with Nimacimab that is not strictly based on food intake. And the most likely effects there are an increase in energy expenditure. And, we don't have great medicines that increase energy expenditure. So if this turns out to be a medicine that will increase energy expenditure, that would very nicely complement the incretins.
That's a good point. Do you have any thoughts on this on the monlunabant kind of difference here? There's the steep drop initially, and the rebound is really interesting, but the difference here in terms of the persistence and consistency of Nimacimab relative to the small molecule.
Yeah, certainly seems like, like I mentioned, it maybe the antibody is just, stuck there for longer, and it takes a while to be removed from the receptor in the peripheral tissue, and for whatever reason, monlunabant gets cleared from that, or maybe there is a stronger negative signal that's imposed by the antibody that is leading to that metabolic rewiring.
Okay. Well, appreciate that, Dr. Gonsalves. I think what we'll do is, in light of the time here, take questions from the audience, and then we can come back to anything else that we didn't cover.
Great, thanks, Puneet. So please hold for a brief moment while we pull for analyst questions. So our first question comes from Mike DiFiore at Evercore. Please go ahead, Mike.
Hi, guys. Thanks so much for taking my question and for also hosting such a great event. Just two for me, and you kind of just preempted my questions in this last segment that you hosted. I guess more clarity on what could explain monlunabant's dose response flattening out. It could just be a matter of native endocannabinoid agonists simply outcompeting monlunabant at the orthosteric site, and then I have a follow-up.
Yeah, Dr. Gonçalves, you want to take that one?
Yes, that is a great point. Yeah, that's completely possible. You know, the fact that this is an allosteric negative site is really to a strong advantage for nimacimab. You know, there is data that they don't actively compete against the endogenous ligand. So I agree that is certainly a possibility, strong possibility.
Okay. And a further clarification on the nimacimab's blunted weight rebound. I mean, it could be due to longer half-life or built-up exposure, but could it also be due to at least a theory that nimacimab is kind of hitting more central peripheral compartments that would cause this metabolic rewiring?
Well, you know, we know the receptors are present in many different tissues, including the adipose and sensory neurons. So, you know, it is possible that you're getting this peripheral effect that is leading to this central rewiring. When I speak of like leptin sensitivity, that's sort of what I mean is those peripheral signals now being integrated and changing the way leptin signals in the brain, increasing its sensitivity. So now when the body's at this lower leptin state, it may feel quote unquote satiated at that lower level.
Okay. Okay. And just one more point of clarification on the upcoming phase 2 readout. Did you say that like over the course of the trial that it would be ideal to have in the combo arm kind of a high teens placebo-adjusted weight loss?
When we're looking at 52 weeks, I think that would be the goal. I think at this point, at 26 weeks, we're looking to see an additive weight loss. So I would say it would probably be double digits is what our target should be at this point. You know, I made a point about really there's no precedent here on a CB1 GLP-1 combo. You know, we have only like CagriSema to look at as a non-incretin potential combo, but that's maybe more of a similar pathway to the GLP-1 pathway. So here, I think showing something in the double digits at 26 weeks would be really something to watch for. The other important thing is watching for the GI tolerability and the slope of the curve at 26 weeks.
Yeah, I would also just you know, caution you if we can use the DIO models to predict, you know, and if you look back at the curves with the tirzepatide plus nimacimab at the early time points, you know, those lines often overlap. So, and you still get efficacy later on. So I think, you know, we should be cautious with interpreting this early data. If, you know, you don't see a big difference between the groups, doesn't mean there will not eventually be a difference.
Yeah, thanks for clarifying that.
Thanks so much, guys.
Thanks, Mike.
Yes, thanks for the questions, Mike. Our next question comes from Ted Tenthoff at Piper Sandler. Please go ahead, Ted.
Great, thanks. Good morning, everyone. And thank you for taking the time to go through this. I particularly appreciate the different scenarios you laid out in terms of where you could potentially develop nimacimab. My question really kind of builds on that. And I'm curious sort of what you guys see as the regulatory path for these different scenarios. And I know it's a little bit early to be asking that. You know, we're about to get the phase 2A data and then obviously the phase 2 studies to run next year. But are there differences that you see, and what kind of flexibility are you going to need just to sort of see how the landscape changes during that time period? Appreciate you taking the question.
Yeah, great. Thank you, Ted. Yeah, I think there's this. I'll direct it over to Dr. Aurora to clarify. But I would just say from the outset here that there's a very clear kind of precedent there in terms of approval path. So we have been focused on that from day one. Is get a drug approved based on monotherapy, but the combo and all of these different things that we're highlighting are still accessible through that regulatory path. And I'll let Dr. Aurora expand on that.
Yeah, thanks, Ted. For bringing that up, I think there is, with the new guidelines, it's quite clear that especially with monotherapy, there is a clear path to approval for these drugs that target weight loss at this point. So, that's how we are looking at it, that being able to establish that nimacimab as a monotherapy is able to cause weight loss is the most direct regulatory path. Having said that, combining it with an agent that's already out there is simply, to my mind, a matter of creating a scenario where you're treating a population that's already taking a drug that's on label and on market.
Right.
So, doing a phase 2 under that scenario and showing the initial data will push us forward as we move forward to the monotherapy regulatory pathway. These are all parallel paths, and sometimes there's an advantage in not being first. You spoke about landscape, and I think as these other agents and combinations start to move forward, it will become clearer exactly what the agency expects from these combination treatments.
Yep.
And Ted, just to clarify, it doesn't preclude us from looking at combinations and maybe aligning with one particular agent and evaluating that as an approval path. So we've continued to be kind of agnostic to combination. We've shown now GLP-1 combo as well as GLP-1 and GIP combo, and we'll continue to evaluate other combinations. But the idea here is to show a development path to any any other agent that might make sense as a regulatory combination.
Great, thank you. Getting excited for CB1 data. Good luck.
Thanks, Ted.
Thanks for the questions, Ted. Our next question comes from Andy Skye at William Blair. Please go ahead, Andy.
Great. Thanks for taking our question. So one thing that Dr. Aurora, you mentioned during the call is the weight loss is not entirely tied to reduced food intake. Really intriguing experiment that you teased out. So I have two parts to this. So one is fatigue. So one thing that patients typically take GLP-1 is fatigue is one adverse event that's really troublesome. And so I'm curious if there's a way that you can tease out from the DIO model about maybe lack of energy from these rodents, and also on the other side, which is increasing energy expenditure from nimacimab. Do you see increased activity in that, or if there is any sort of quantifiable way to measure that, just to get a sense of what the phase 2b readout would look like? And then maybe bigger picture questions as we kind of think about maintenance. I'm curious, from the physicians, for those who stay on GLP-1s for longer term, so beyond maybe the 1 year mark or 2 year mark, what are some characteristics do you typically see from these group of long-term GLP-1 users? So that's second part. And the third part, I think, now we've seen maybe data from all three modalities, right? So the peptides, the small molecules, and antibodies. One thing that we saw from MariTide is that the slope beyond 52 weeks is actually still kind of a straight line. And nimacimab is an antibody. So I'm just curious if that's is it kind of out of the realm of possibility that you might see kind of MariTide-like slope as you look at it from a longer term perspective. Thank you.
Yeah, thanks, Andy. I'll address the first part of this. In fact, Marcus can join in there. He actually has great expertise in this domain. We do ask our partners to look at these mice carefully because there is some evidence, even with rimonabant, there was a way early on to see some behavioral changes, etc., in early experiments. And to date, at least, we've never had any reports of actually seeing any issues with in any of our animal cohorts. And these animals seem to behave well and seem to have good energy and don't seem to show any problems. But as I said, Marcus is the expert here, so I'm going to hand it over to him.
Yeah, happy to speak about this for hours and hours, but I'll be brief. So, typically what happens in any scenario of weight loss is the energy expenditure goes down. And so if this agent were able to compensate that mechanism, that's a way that the body tries to preserve calories and enhance weight regain. So it seems likely based on the ParaFed data that nimacimab is increasing energy expenditure. And I think the team here is very eager to get those measures directly. You could do indirect calorimetry on mice, and you can actually prove that their total energy expenditure is going up, or at least not dropping as low as the incretins. And it's intuitive for us to link that energy expenditure with physical activity, but in rodents, they are very efficient movers, and changes in physical activity don't always correlate with changes in total energy expenditure. But it is a good surrogate for fatigue, as you mentioned. So I think if we accurately measure the physical activity and total energy expenditure, it will help address your main 2 points, which are, is there any fatigue, and is there any changes in the total energy expenditure?
The second question is around the long-term maintenance. Dr. Cannon, Dr. Warden, do you want to take that?
Yeah, so one of the questions were why do people stay on the medication? So someone who's on it for longer than a year, who are they? Why are they on it versus the rest of the people who have actually stopped it? The answer is I don't know. And we don't know. What we do sometimes see is that the person who is most knowledgeable about the medication has a really good understanding of why they're taking it for something beyond weight change, for anything from their osteoarthritis, their rheumatoid arthritis, their joints get better, their body gets better, they're thinking about Alzheimer's risk. They have and frequently it also has impact on in terms of mood. So what we've researched and showed is that somebody who has a good mood before they start GLP-1s has a similar good mood after they end up starting it. If they have a bad mood, then it's bad at the end too. There's no change. There's no fluctuation in that. So what I'm saying is that if you've got a good attitude coming in, you will continue to have that good attitude and likely stay on the medication. If there's a lot of negativities about your whole life in the first place, this isn't necessarily going to fix it. It fixes a couple of things, but it doesn't fix your challenged life. And those are the people who tend to stop the medications because their whole life is challenging. And so, but of course, this medication is going to go to these medications go to millions of people. So you get everybody. You get those who stay on and those who don't, and we have to deal with both people and understand how we can manage both of those type of people.
I mean, I also think it's very important, and this is also a prolonged discussion as well, to not treat all weight loss patients as the same, not monofaceted. I mean, you have the midlife population that have kind of gotten off track in their late 30s, mid 40s, that just kind of need a reset button. You have the over 50 population, the postmenopausal population that have been living with obesity for a much more prolonged period that need, I believe, a much more prolonged maintenance therapy. And again, again, it's not monofaceted as far as why people drop off medicine, whether it's cost prohibitive, whether it's GI side effect. But again, I agree with Dr. Wharton that the more educated the patient is of why they're taking what they're taking, allows them to do it, to stay on for a prolonged period of time. And again, this medicine plays into that as far as with better GI tolerability, all the stuff that we've already hit, preservation of lean muscle mass, quality of life, that is setting this up for success with this medicine. I wholeheartedly believe that.
Andy, thank you, Dr. Cannon and Dr. Warden. Andy, what was the third question?
Yeah, pharmacology, antibody, longer, potential, kind of tail to the weight loss curve.
Yeah. So in terms of the half-life and PK/PD profile, is that what you're referring to? Or what's
Oh, sorry. So basically, we saw three different modalities, right? The small molecules, the
oh
peptides, the antibody. So just curious, right? Maritide, you basically see kind of a linear trajectory over 52 weeks, and I'm just curious about nimacimab. Is it unreasonable to expect that kind of weight loss curve as you think about longer term or the extension study that's reading out 2026?
yeah, it's an interesting question. Do you want to take that one, Dr. Aurora?
Yeah, Andy, maltide is a really interesting molecule for more reasons than one. You pointed out it's an antibody, which is really encouraging for us because people have often made this argument the other way that antibodies will not work. They cannot penetrate to the right place. They don't go to the hypothalamic receptors. And I think here you have some proof of concept that none of that is necessarily true, and what the hypothalamus perceives in terms of what your energy intake is and how that balance is created may well be influenced as much by an antibody without really crossing the blood-brain barrier as you see with a small molecule or with a peptide. So I think it's intriguing in that sense too. Here there is this question of a bit of a compartment model, which may or may not apply to maritide, but certainly in the case of nimacimab, we question, especially looking at these high doses as we keep escalating doses in mice and finding greater effect, the question of whether we are just getting more drug into peripheral compartments that are important, like adipose tissue, and then they're staying there. The drug is staying there and having a longer term effect. So we hope that will translate into longer efficacy and just more durable efficacy. And that may be an antibody-mediated mechanism compared to small molecules.
Helpful. Thank you so much.
Great. Thanks for the questions, Andy. Our next question comes from Jay Olson at Oppenheimer. Please go ahead, Jay.
Hey guys, thanks for providing this comprehensive educational webcast. We have a few questions. First, based on these impressive DIO mouse model findings, any thoughts on the potential for lower and/or less frequent dosing of nimacimab for weight loss maintenance versus weight loss induction?
Yeah, so I'll take the first part of that, and we can maybe Dr. Aurora, you can talk about it from a development standpoint. So we are in parallel continuing to look at both from CMC in terms of increasing the concentration, and in terms of from a clinical perspective, the idea in our next phase 2b is to evaluate dose frequency and dose and also additional doses, something we weren't able to do in the proof of concept study. So the idea here is to get to a TPP that would be a less frequent dosing than a weekly injection. Our ideal TPP is the monthly injection, which is what we're working towards ahead of phase 3.
Okay, great.
Okay, so our goal is to move on to phase 2b, which would be a definitive dose ranging study because we want to pick the optimal dose to move on to phase 3 or pivotal study. And so as part of that, we are interested in both what is the right dose, because we're learning as we go along that there may be a dose range to be done here, and also what is the most appropriate frequency combined with that dose. And so yes, we are very interested in that. And as Puneet mentioned, as we work with formulations and be able to concentrate the molecule better and get a concentrated formulation, it makes it that much easier for us to work with a decreased frequency of administration. So yes, that's certainly something we'll be looking at.
Okay, thank you. And then given your latest preclinical data supporting post-tirzepatide maintenance and the high unmet need for post-GLP-1 weight rebound control, how are you thinking about the weight loss maintenance commercial opportunity for nimacimab, and recognizing that we're still waiting for regulatory guidance on how to achieve a weight loss maintenance indication? Is that a market or indication that you're planning to pursue?
I'm gonna make a really bullish remark, and I'll say that's a sweet spot for nimacimab in terms of being an opportunity for the taking, especially if we continue to highlight a really tolerable profile, and that's where we're looking forward to seeing that. We've been highlighting it partially also from the standpoint that we've done an extensive amount of work with clinician work this year and understanding what that market is, recognizing that about 70% of patients are discontinuing within a year, 80% within 2 years. We've already talked about the relevance of that market, but the idea here is that we don't think it's a requirement to change the regulatory the strategy that we have at the moment. As a second line option, if you will, it's still a tremendous market opportunity. And the way that we look at maintenance is that there's an opportunity of non-responders in the primary setting, in the first line setting, and then there's a rescue population, the patients that come off of tirzepatide, patients that come off of semaglutide, or any other of the approved AOMs and are looking for an alternative. So there's a pretty large market based on our modeling, and we feel confident about being able to really secure that market.
Great, that's super helpful. Thanks. And then maybe one last question from us. Since there is some evidence showing that weight loss with tirzepatide has superior body composition to semaglutide in terms of less lean mass loss, are there any insights into weight loss composition for the tirzepatide nimacimab combo and how that compares to tirzepatide alone?
Yeah, there was a more comprehensive set of information in the slides. I don't have the slides up right now, and it's not going to be easy to refer to. But Dr. Rohr, do you want to? We basically had to answer your question, Jay. It was biased towards lean mass preservation, so where we are targeting fat. Whether there's a difference between tirzepatide and semaglutide, I don't think we have that direct comparison. We'd have to look at both of those studies individually. But the bias is towards targeting fat.
Great. Thanks for taking all the questions.
Yeah, there's all the side-by-side stuff is available, and it'll be available after this call in terms of all the slides, so we can take that offline. There's another question that was from Oppenheimer in the audience panel, and it was, what gives you confidence in human dose of 200 mg in your phase 2A in comparison to mouse dose of 240 mg per kg? So I think from our PK modeling, just to answer that question, is that we feel confident that the 200 mg is equivalent to our 75 mg dose, and the preclinical data kind of highlights in those curves that even the dose-dependent weight loss is pretty close when you look at 75 and 240. So we feel very confident with the 200 mg dose that we have in the clinic at the moment.
Great. Thanks again for taking the questions.
Thanks, Jay. Our next question comes from John Wolleben at Citizens. Please go ahead, John.
Hey, good morning, and thanks for hosting this event this morning. Very helpful. Just wanted to dig in a little bit on GI tolerability. You guys gave us some benchmarks for the other metrics you're looking for, and just wondering if there's specific thresholds you want to see here to say, hey, this is better than the incretins or not. And is it as simple as just looking at the Wegovy arm, or are there cross-trial comparisons we should be making to other agents? And just piggybacking on that, do you expect to see improved tolerability for the combo arm versus Wegovy alone? So any color you can provide on tolerability benchmarks would be helpful.
Sure. I'll just set the stage for benchmarks. The CB1 benchmark is about 30% GI disturbance in the small molecule setting. So if you look at rimonabant data, which is the largest data set, that was about 30%. And then also, I think in the monlunabant data in the phase 2, that was reported. Dr. Warden, correct me if I'm wrong on that. On the GLP-1 side, that's been substantially higher. So our expectation is to come under those numbers. So to have less than 30% GI disturbance. But if we use phase 1 as a proxy, even though it was a smaller duration study, that was a very clean profile with a very limited, I think a few mild GI adverse events. Sorry, Dr. Warden, I think you may want.
No, that's right. That's correct. I mean, and the question also was partially whether it'll just be looking at the GLP-1s like a monotherapy GLP-1, which is what the semaglutide is. But looking at there may be other molecules that are out there, a combination of a GLP-1 and an amylin, or amylin dose by itself. But really, one of the and what the GI side effects are there and what the comparisons are. So who you're comparing it to, and when this comes to market, what will be the necessary comparison. I think that one of the big positions is that people should be looking at, and of course Skye is actually looking at, Skye Bioscience looking at this, is smaller doses of 2 molecules to get the best GI tolerability possible. That's become a big thing in the market. This is not the only company that's looking at that, because it's important. It's important to try and see, can we get low dose of one molecule that therefore then the GI tolerance is very low, another low dose of another medication, GI tolerance really good, and then be able to combine those together, have a magnified efficacy with the still continued low GI adverse effect profile. That's the ideal, and that's of course what Skye Bioscience is trying to do here, and we cross our fingers that it actually happens. But it's not an unreasonable thing to try to do.
Puneet, what's your definition of GI disturbance, just so we're on the same page?
It's just the 3 that we've been looking at is nausea, vomiting, and diarrhea.
Yeah, those
overall rate of those 3.
Yeah, those are the 3 we focus on, because that's what we see a lot with the GLP-1s. It's also other things like dry mouth and abdominal distension that you see with a variety of agents. So when you hear the total numbers, like a 30%, it usually means all GI-related adverse events. But yeah, the ones we're mostly focused on are nausea, vomiting, diarrhea, which are most troublesome to patients.
Got it. Okay, very helpful. Thanks for taking the questions, guys.
Yeah, nausea has really stood out in our survey of speaking to physicians. Is that's one of the leading issues that patients are dealing with.
Another measure from the studies that I really value is the discontinuation rates due to the GI AEs. It hovers around like 4 to 6%, depending on the study.
Yeah, that's a good point.
Great. Thanks for the questions, John. So our final question comes from Albert Lowe at Craig-Hallum. Please go ahead, Albert.
Thanks for taking my question. The first one for the KOLs on the panel. I was wondering if you can speak on what portion of your patients are non-responders to GLP-1s. Maybe similarly, what portion of patients require additional weight loss beyond what they can get with GLP-1s?
Kevin, you want to add? Yeah. So in terms of the non-responders, it was a lot higher when we were looking at the initial GLP-1 analogs. As we introduce more and more, it becomes fewer and fewer. So it's we sometimes almost ask whether they're actually doing the injection, or they're injecting it into the air, because we sometimes just don't believe that you can have a non-responder with some of the newer molecules we have with such high rates. So it's not huge numbers, but what the bigger numbers are people who didn't get what they wanted, and that's important. It's like, I was looking for this and I'm going to keep on looking for it, and I don't think this medication is working well enough for me in terms of the price I'm actually paying for. It's not worth the value. So I want something more. That something more could be actually combining to something else or switching over to something else. So I think that's more so where some of the bigger questions are. Because what we're seeing is that those that have greater than 5% weight loss with some of the newer agents, it's 100% of patients have greater than 5%. But if somebody only lands at 6% weight loss with that medication, they may not want to deal with either the side effects or the cost of it, and say, I want more. So those are the bigger patients, and that's a a decent number. I would say in the range of about 15 to 20% of our patients. And the second question was about what number of patients?
That would maybe require additional weight loss. I guess maybe perhaps you answered it maybe within that. But maybe
Yeah. I think a big section that we need to continue to focus on is not those who require so much weight loss, right? Those are surgical patients. BMI is a 50, 60, 70. We still have those, and those do come down. And frequently, it is again about how much benefit they get from the other things that they actually need. So I don't always think of that population as the population that drives either the market or the financial components. The population that drives it is obesity class 1, and even those who are in the overweight range of 27 to 30. That's the market that we should frequently think about and focus on. They're a big market, and they're sometimes forgotten. We think about the BMIs of 50 patients, but that smaller BMI is a huge number of patients.
Yeah, I agree. I mean, we have kind of a disproportionate population in the sense of all of our patients that enter into this program work hand in hand with a dietitian who is extremely effective. So we actually typically will see that greater percent weight loss, but I have to keep in mind that there's a much larger percentage of the population that is simply being prescribed these medicines. And, we are able to do risk mitigation steps with our dietitian to kind of set them up for success. But again, like Dr. Wharton said, there's a much larger population that will simply be prescribed these medicines that, as long as we are able to make them more GI tolerable and more effective, they are going to stay on maintenance a lot better. So again, my population is a little bit skewed because, again, I see it through the lens of working hand in hand with this dietitian that does a phenomenal job. But again, I have to keep in mind that a much, much, much larger percentage of the population wants kind of more that simple, easy button that if we can strive to get that for them, it's going to be very effective and very well tolerated and very, a home run, basically.
Thanks. Maybe along those lines of what patients want, I guess maybe how much demand or what are there patients that you think would prefer nimacimab perhaps ahead of a GLP-1 because of the advantages in preserving muscle mass despite perhaps less overall weight loss as a monotherapy setting?
My thought process is skewed in that direction. So yes, but again, with that's going to be from a more educated, larger population. Again, patients by and large, they just look at the number and the percentage weight loss. As more and more evidence comes out and more information comes out, it is going to become more common knowledge that a quality of life aspect is going to be significantly related to that muscle preservation. And again, that's going to happen over time. I'm convinced of that.
All right, great. Thanks again for taking the question, and looking forward to seeing the data here soon.
Thanks, Albert. Tara, I think that wraps it up for today. We've definitely gone over our allotted time.
Yes, that's all the time we have. So thank you everyone for joining. You may now disconnect.